today. So she asked me to introduce our speaker. And I see that we have a number of folks from outside the department, which is great. So maybe I'll give my usual spiel about the a master's thesis defense, which is what is happening today. Angie McGraw is our speaker. She will be defending her thesis. I assume her committee is here. She'll give a public presentation. We'll take any questions from the public, and then the public will be dismissed, and her committee may have further questions for her, and then she'll be dismissed, and the committee will decide. So just a few comments about Angie, who's probably known to most of you. She is about to complete, pending this defense, her Master of Science in the Bioinformatics and Computational Biomedicine major. And she actually is already in a job. She is currently in the NIH Post-Baccalaureate Intramural Research Training Award Program in the lab of Dr. Nanping Peter Wang, who I see here. Before coming to OHSU, Angie received a Bachelor of Science in Computer Science from Portland State. And so I will turn it over to her.
All right, great. Thank you so much for the introduction. My name is Angie McGraw, and the title of my thesis is An Evaluation of Best Practices for Clinical Laboratory Improvement Amendments, Bioinformatics Pipelines. As for a little bit of a premise, given the rapid advances in technology and computational complexity, there may be gaps in best practices and guidelines to support the current challenges facing CLIA laboratories. For our study overview, next-generation sequencing technologies have provided opportunities for personalized treatments. Whole genome sequencing, which is an NGS technology, allows for the exponential generation of data. We highlight the potential of NGS-based bioinformatics pipelines and precision breast oncology treatments and the need for standardization. Broadly speaking, we need set guidelines and standards to ensure pipeline accuracy, appropriateness, and efficacy, as well as those to protect patient safety and ensure quality patient care. Our study has two aims. The first was to conduct a literature review to assess current best practices for CLIA bioinformatics pipelines and to identify potential gaps. Our second aim, the use case for our second aim, is to determine reference background distributions for expression signatures for clinical usage. So now we're going to be diving into aim one. All right, so Lax Laboratories. This was a headline newspaper article in the Wall Street Journal. And up until this point, so we're thinking about late 1900s, up until this point, laboratories had been largely unregulated. And this illustrates one example of that, in which people were getting incorrect results with cancer screening. And so essentially, people were being told that they had cancer when they didn't have cancer, and this is an indication of a false positive. And this whole situation indicated why there needed to be more guidance and regulations when it comes to clinical laboratories. So in response to concerns regarding clinical laboratory testing, CLIA was established in 1988. They stated that certification certifies that the laboratory has developed a validated clinical assay that is reproducible, precise, and accurate, with established sensitivity and specificity. They also state that laboratories must maintain data management systems, entail accurate record keeping, and implement standards for bioinformatics pipelines. So this timeline is not an exhaustive timeline, but it indicates how laboratory testing has evolved over time. So starting off from the left-hand side, we're looking at 1993 and earlier. So these were your ELISA or antigen-based tests. So these were simpler tests. And so moving on from 1993, we saw that laboratory testing has evolved to include omics-based tests. And what omics basically means is that when you have sequencing data that falls under the realm of omics, and with omics, you have bioinformatics because you need a way to process the sequencing data. So what this essentially means is that bioinformatics is going to be here to stay because we're constantly generating sequencing data. And so in 2006, there was draft guidance on IVD MIAs, and these were in vitro diagnostic multivariate index assays. And so this was the very first time where we had clinical data, an algorithm, and a threshold to interpret the results. So the first IVD MIA assay that was approved was MammaPrint. And so MammaPrint was a 70-gene microarray-based test. And what MammaPrint does is that it predicts whether breast cancer is likely to return within 5 to 10 years. And since then, since the approval of this assay, there's been more NGS-based mutation and gene signatures, as well as other new tests. And so now we're really getting into this idea of clinical genomics. So clinical genomics has become really pervasive, especially in the recent years. And so for the sake of time, we're going to be focused on the right-hand side of this diagram. But what this diagram essentially shows is that overall, clinical genomics is a dynamic process because annotations are constantly being updated. And so for the right-hand side, a common use case for clinical genomics is genetic screening. And with genetic screening, we're looking at NGS, which again is next generation sequencing. So we're looking at either whole exome sequencing, whole genome sequencing, or a targeted array. And so the pipelines are now going to be focused on alignment, variant calling, and filtering, as well as classification. And so to dive further into clinical genomics, we conducted a literature review. So in this literature review, in this top panel, so this top graph right here, we're going to be looking at clinical bioinformatics and clinical genomics. And in this, we see that clinical genomics gives us a greater span than clinical bioinformatics. But then when we looked at this bottom panel here, we looked at medical genetics, and we saw that medical genetics provided a larger and more relevant corpus for a review. And so it's important to note that these y-axes aren't to the same scale. The top panel caps out around 2,000, and then the bottom panel caps out around 20,000, which means that medical genetics has become more prevalent in research. We also found through our literature review that there's high variability in applications when it comes to medical genetics. So for instance, in clinical usage, potential for clinical usage in CLIA, and so on. So continuing with our timeline. So this is 2012. This is just 5 years from when that first assay that incorporated clinical data and algorithm and threshold, that was just 5 years from when this assay was approved. So just 5 years from this, there was scientific fraud and falsification that occurred. So there was an Institute of Medicine report on translational omics. And so this report came about because there was scientific fraud and falsification that resulted in incorrect gene expression-based signatures used in clinical cancer trials on patients. So the researchers claimed that they've achieved a genomic technology that could predict with up to 90% accuracy which early stage lung cancer patients were likely to have a recurrence and would therefore benefit from chemotherapy. And so the data that they were using was falsified. And so they were intentionally misassigning the patients as to like whether or not they would benefit from chemotherapy or not. And so this report from the Institute of Medicine focused on how to prevent this from happening again. So this report was followed up in 2013 by more guidelines from the National Cancer Institute with omics-based test development scientists. And so these guidelines apply to any clinical trial involving the use of an omics test, which is a sequencing test that would involve the clinical management of patients in a given trial. And so that means like any tests that involve the selection of therapy for a patient. And so there were a number of recommendations, but this slide just highlights those on model development. And many of these recommendations were involving increased rigor and how algorithms and methods needed to be more appropriate. In 2014, so a year after, we had a technology review that highlighted the rapid shift towards clinical sequencing. And when we're reading this figure, each of these rows or each of these bars indicates a company. So 29 would mean that there's 29 companies in this topic or this category of consumables and arrays. And so for all purposes, we're going to be focused on this red box right here. And this red box includes alignment and annotation, interpretation and reporting, and data storage and bundling. So what this figure essentially shows is that companies were providing arrays and sequencing, but then they moved towards providing bioinformatics support, so a way to process the sequencing that they're generating. And so I'd now like to highlight 2 case studies that involve bioinformatics workflows. So this first case study highlights how to address the issue of increasing rigor and reducing error in pipelines. So this first case study is titled The Use of Semantic Workflows to Enhance Transparency and Reproducibility in Clinical Omics. So semantic workflows, in simple terms, this is where users are guided through each workflow run. So there's going to be guidelines for the user to follow, as well as interpretations of the workflow. And so this type of workflow, semantic workflows, enhance analytical validity and patient safety. So in the study, they stated that workflow platforms such as Galaxy and Taverna have increased the use, transparency, and reproducibility of omics analysis pipelines. They also state that domain knowledge creates challenges when attempting to transfer a pipeline from research to clinical settings. As a basic study overview, they analyzed the effectiveness of semantic workflows, and the workflows that they analyzed was called WINGS. They also implemented a clinical omics pipeline to annotate DNA sequence variants identified through NGS technologies. So one of the key findings was that implementation of a clinical omics pipeline through a semantic workflow allowed for the pipeline to meet the clinical omics standards for transparency, reproducibility, and analytical validity. The system that they analyzed addresses 4 needs in clinical omics analyses, one being frequent updates to molecular life science databases. Science is constantly being done, which means that these databases are frequently being updated. 2 being that heterogeneity and consistency of biological data. 3 being rapid development of omics software tools. So with this exponential generation of data, people are going to try to be able to tackle this data. So that's why the development process is going to be rapid. And then 4, processing of large omics data sets. They also included a checklist for pipeline development. So they said that pipeline development, they should include exact input data used for the analysis, key intermediate data generated from the analysis, third-party data, and then all code and software used in the analysis. And these 4 checklist points indicate that transparency and reproducibility contributes to increasing rigor and reducing errors in pipelines. The second case study is a study examining an implemented pipeline for pathogen identification. And this is a public health study. So this study is called a cloud-compatible bioinformatics pipeline for ultra-rapid pathogen identification from next-generation sequencing of clinical samples. So they stated that NGS technologies have allowed for development of computational approaches to maintain public health. So there's high applicability with bioinformatics pipelines, but there's difficulties to transition from a research setting to clinical trials because of integration, accuracy, and time efficiency. As a basic study overview, they utilized metagenomic data from 237 clinical samples, and from this, they constructed a cloud-compatible bioinformatics pipeline called CERPI for comprehensive pathogen identification. They identified 3 main challenges with developing bioinformatics pipelines. One being that alignment and classification algorithms must contend with massive amounts of sequence data. 2 being that only a small fraction of short NGS reads in clinical metagenomic data corresponds to the pathogens. And 3 being that novel microorganisms with divergent genomes are not well represented in existing reference databases. They also assessed CERPI's feasibility in real-time clinical analysis. So they utilized acute serum sample from a 20-year-old female with a 3-day fever, myalgias, and a headache. This patient was exposed to a region of Australia that had mosquito-borne alpha viruses. And within 13 minutes, sequences that spanned the HHV7 virus were detected. And so the fact illustrated here is that you could have a very high false negative rate where only a small amount of the sequences are actual variants. And so a big challenge in this use case is that false negatives are a huge issue. And so the prior use case that we went to faces the challenge of how to reduce error, whereas this case study faces the challenge of reducing false negatives. And so as a takeaway from this case study, the pipeline that they analyzed was both accurate and efficient and was able to produce results in the desired time frame. So now moving forward with our timeline, we're in 2018 now. This is where we had joint recommendations from the Association for Molecular Pathology and the College of American Pathologists. They stated that because there's a lack of published guidance, there's currently a high degree of variability in how members of the global molecular genetics and pathology community establish and validate bioinformatics pipelines. So before, CLIA came in to provide guidelines, but tests, as you can imagine, over time become more complex and more variable. And people assumed that CLIA would cover every aspect of pipeline development, but since tests became so variable and so complex so rapidly, these guidelines don't cover every aspect of the bioinformatics pipeline development process. And so these joint recommendations involve 17 best practices, and the key themes were training and validation. And we want to place an emphasis that when it comes to training, training isn't going to be a shortcoming of the trainer or the learner, but rather more training alone isn't going to solve the problems. We're going to need to think about what needs to be included in the training, and we need to make sure that the training is obligated. So we updated our literature review to being since 2018. So 2018 was when we had the joint recommendations. So then we carried out a literature review from 2018 and onwards. And the three key terms that we were focused on were validation, bioinformatics, and clinical. And as you can see, over the years, we've seen an increase in these three terms in literature, indicating their importance in literature, their increasing importance in literature. And so from the literature review, we saw that there are still challenges in validation. And currently, with existing methods, they involve case studies and use case reports. And then in some instances, publications can be used as a surrogate for actual validation, meaning some publications that are producing a pipeline will be using other publications to validate their data instead of having parameters and models to test their pipelines. And then moving to this middle box here, there's also no pre-existing methods or workflows, so there's no baseline comparison point for the pipelines. In addition, level of validation for algorithms vary, and there's also a lack of a gold standard and appropriate references. And then in terms of artificial intelligence and machine learning, there's a lack of explainability and interpretability, and especially for deep learning. There's also training versus operational site differences, and then we're also seeing distributional drifts, so a drift in the data, which can also impact the outcome of the model. So the lack of a gold standard and a reference was the area that we had the opportunity to investigate in AIM-2. So AIM-2, our use case for this was to determine background distributions for expression signatures for clinical usage. So starting off, one of the key things here is to understand what an ideal reference or null distribution is. So the goal here is to have an appropriate reference distribution, so a baseline comparison point. So this distribution would give us appropriateness to assess outliers due to real treatment effects. So thinking back to basic statistics, we have a normal distribution curve on the screen. And so this normal distribution curve, at the extreme tails, the ends of this would be a patient sample, indicating actual treatment effect. So we stated we had 4 key characteristics for an optimal reference or null distribution. One being that expressions in samples are appropriate for the disease type. So our reference distribution should consist of gene expressions that are appropriate for the given disease that we're looking at. 2 being that there's biological variability sample to sample, but isn't due to technical artifacts. So there's obviously going to be differences in gene expressions, but this should be due because of the differences in samples and not due to differences in batches. Thirdly, there should be a large enough distribution to avoid sampling error. So the genes that we're looking at, there should be enough relevant genes for us to avoid sampling error. And fourth, pairwise differences among samples should reflect normal biological variability, but be less than the actual treatment effects at present. And this would allow us to identify real changes on therapy. So the measure that we were looking at, or the samples that we're looking at for our distribution, for our null reference distribution, are UHR replicates. And so UHR replicates, essentially, they're RNA isolated from a diverse set of 10 cancer cell lines. And what a cancer cell line is that they're cancer cells that keep dividing and growing over time, and normally they're in a lab condition. And so these are a collection of cells originating from one cells. So the pros of using UHR replicates for a reference distribution is that they're a universal reference. They're also, again, a mixture of cancer cell lines. And from literature, we know that they provide background distributions to assess significant pre- and post-treatment changes. The cons of using UHR replicates is that even though they're a mixture of cancer cell lines, they're not going to be exhaustive for all cell cancer types. They're also lab-pooled samples and not actual patient samples. And our use case for this was looking at deltas and rank deltas. So both of them measured differences in gene expressions for patients pre and on treatment. So as a little bit of background on breast cancer, which is the disease that we're going to be looking at for our analyses, is that breast cancer is the leading cause of cancer-related deaths among women in the United States. Breast tumors develop into benign tumors or metastatic carcinomas through constant stimulation. And currently, the 5-year survival rate is above 80% due to early prevention. And there's approximately 2.2 million newly diagnosed cases and over 500,000 death cases that were reported in 2020. So breast cancer is currently curable for approximately 70 to 80% of patients with the early non-metastatic stage of breast cancer. But when we move to more invasive cancers, that's where treatment becomes more difficult. So now looking at current breast cancer therapy. So we have non-metastatic cancer. So this is where cancer is localized to the origin point. And then we have metastatic cancer, where cancer is spread from the origin point to other organs. So the more invasive cancer. So for non-metastatic cancer, the goal of the therapy is to eradicate the tumor from the breast and regional lymph nodes, and to also prevent metastatic recurrence. Current therapies involve surgical resection, sampling or removal of axillary lymph nodes, and post-operative radiation. For metastatic cancer, this is treatment based upon metastatic cancer subtypes. The goal of this therapy is to prolong the patient's life and to provide relief to the patient. And for therapies for metastatic cancer, we're going to be targeting multiple cells or tissues with receptors for a particular drug. So now we're moving towards precision oncology. And precision oncology follows the idea that since each patient is unique, their cancer is going to be unique. And precision oncology utilizes a patient's molecular and biological features to optimize therapy and drugs that target oncogenic mechanisms. And as outlined by a recent study, many treatments have been designed for patients with the same disease, but the following might be observed. 1, symptoms of disease may be reduced, and this is what we want. 2 and 3, expected responses may fail to occur and side effects may arise, which is what we don't want. And so now with precision oncology, we're going to be moving towards studying gene expression in specific organs, tissues, or tumors. So then that means we're going to be looking at understanding the differences in gene expression between patients and then pre and on therapy. So the hallmarks of cancer, these are essential to cancer biology. So hallmarks of cancer are going to be essentially 6 traits that define cancers. So the first one is sustaining proliferative signaling. The second one is evading growth suppressors. Third one is activating invasion and metastasis. Fourth is enabling replicative immortality. Fifth is inducing angiogenesis. And 6th is resisting cell death. So the purpose of using the hallmarks is to see if certain hallmarks are unregulated in a given patient. And then this is important because there are 50 gene sets that represent the hallmarks, but the hallmarks are comprised of 6 traits. So for our purposes, we're looking at 27 of the 50 gene sets. And these 27 gene sets were prioritized by the Knight Diagnostic Laboratories. And there's 27 of them, but I'll just list out a couple. One of them is estrogen response early, and then another is MYC targets version 1. So there's been an increasing interest in the application of the hallmarks. So the study on the left in 2018 was titled The Diverse Genomic Landscape of Clinically Low-Risk Prostate Cancer. So they observed tumor biology and gene expression patterns among men with clinically low-risk prostate cancer. And they utilized hallmark gene sets such as MYC targets, G2M checkpoints, and E2F targets. And in this study, they determined average genomic risk scores from the hallmark gene sets. And they found that risk scores were associated with worse pathology findings, and they were associated with prostate-specific antigen recurrence after surgery. In the study on the right, which is in 2020, this is titled The Interrogation of Dysregulated Pathways Enables Precision Medicine in Mantle Cell Lymphoma. What they did was that they conducted molecular profiling analysis on clinical patient samples and identified hallmarks such as the aberrant apoptosis and E2F targets in the patient samples. So from their study, they identified 3 enriched pathways in relapsed mantle cell lymphoma from the hallmarks that they analyzed. And so the takeaway from the increasing interest is that changes in the hallmarks are thought to provide insight in terms of patient response. So in our study, we assessed whether or not UHR replicates would be an appropriate reference distribution. The UHRs, again, are composed of various cancer cell lines. And so the genes associated with the UHRs were filtered by the prioritized 27 gene sets for breast cancer. We also had de-identified patient data, and these are paired. We had pre and on treatment. And in addition, of the 42 UHR replicates, there are 2 different lots based upon sequencing dates. So from the 42, we went down to 38 UHR samples based on the sequencing dates. Lot 1 was sequencing conducted after November 9th, 2019. Lot 2 was sequencing conducted before August 22nd, 2018. And for both of these lots, 1 large fragmentation batch was prepared. And then lot 1, the batch has been used for all subsequent sequencing runs. So now we can take a look at the average expression of the data before filtering. And so as we can see here, the y-axis represents the logged average expression. The x-axis is labeled out by the number of UHRs we had, and then they're labeled out by what lot they came from. So we had 38 UHRs. And so looking at this, we can see that the medians for the box plots are relatively similar. However, the most concerning part about this box plot is how long the tails are and how much variability that these tails indicate. And so because of this variability, we wanted to look towards filtering in order to tackle this variability. So then that leads us into our workflow. So first, we would evaluate gene expression and compare filterings. So we had two filtering methods. One is more stringent, the other one is less stringent. Then we would compute differences in gene expressions for the UHR distribution. So differences in gene expressions, we called them deltas, and then the differences would be the absolute value of the differences. Then we computed the differences in gene expressions for the patient distribution. And so the patient distribution would be used as a comparison point for our UHR distribution. Then we would compare the UHR distribution with the patient distribution. And as we hypothesized, the UHR expressions should be representative of the patient samples. Then lastly, we would evaluate the UHR as a null reference distribution. So basically an assessment of whether or not the UHR distribution meets the key characteristics for an optimal reference or null distribution. In addition, we want to see if we can generalize this with other cancers, not just breast cancer. So now we're looking at the move towards filtering. As we saw in the unfiltered box plots, we saw long tails indicating high variability. And when comparing it with this patient box plot here, we can see that the patients have shorter tails in terms of this area right here. And so this indicates that the UHR distribution had more variability than the patient distribution. And so this patient distribution, again, was the 2D identified patient samples with pre and on therapy values. And what's interesting here is that when we're looking at the median, the UHR distribution had a median of 3.1, whereas the patient distribution had a median of 8.9. And so since we saw variability between lots for the UHR distribution, and in addition, higher variability when lot 1 and lot 2 were combined versus separate, we also saw a large distribution of low expressing genes. And then from this, we had two sets of cutoff metrics, which again, the first one is more stringent, and then the second one is less stringent. So going further in depth with our cutoff metrics, for set 1 and set 2, we start out with 42 EHRs. And the reason why 42 goes down to 38 is because, again, the sequencing dates that we were interested in looking at, both of the sets started out with 60,000 genes. However, set 1 went down to 533 genes, being a more stringent filtering criteria. And then set 2 went from 60,000 to 10,000, being less stringent than the first set of cutoff metrics. And so for lot 1, we had 26 UHRs, giving us 325 pairwise deltas for a given gene. And it's basically 26 choose 2. And then similarly with lot 2, and then the overall pairwise deltas. So now we can take a look at the average expression values for the first set of cutoff metrics. And as we can see, the tails are a lot shorter, but there's still variability within this area here. We can also see that the medians are relatively similar, which is what we saw before filtering. And so with this first set of cutoff metrics, we went from 60,000 to 533. So we have fewer genes, but the overall expression is higher, and we had more modest deltas with this cutoff metric. We can also look at the second set of cutoff metrics. So we went from 60,000 to 10,000, so more genes, but then we have lower expression and higher variability as indicated by these tails here. So the evaluation of utilizing ranks. So we have deltas, which is just differences based upon gene expression. These were two variable because we saw batch effects between the lots, so lot 1 and lot 2, and then we also had scaling issues. And then we moved on to using rank deltas. So this would be determining ranks based on gene expression. The highest gene expression would receive a rank of 1, second highest would receive a rank of 2. And so then we would take the differences based upon the ranks. And so this would tackle the issue of the scaling. So the key question that we wanted to answer with the rank deltas was that, are the genes with no or low expression in the UHRs affecting the distribution of the ranks? So first, we filtered the genes to select the genes that were associated with 27 gene sets that represented the hallmarks. And so our filtering thresholds for the UHRs, there's 4 of them. 1 is keeping genes with 0 expression. 2 is removing genes with 0 expression. 3 is keeping genes with greater than 1 average expression, and 4 is keeping genes with greater than 10 average expression. So as for our methods, for the UHR distribution, we removed genes based on the filtering evaluation. So those were the 4 filtering thresholds that we saw on the previous slide. We then extracted the genes associated with each of the 27 gene sets that were associated with the hallmarks. So from there, we had 27 data frames that we were working with. And we also had a patient comparator, which is the two de-identified patient samples, both with biopsy 1, so pre-treatment, and then biopsy 2 being on treatment. From there, we computed the ranks. So a higher gene expression would be a lower rank number. Then we computed the rank deltas by taking the absolute value of the differences. And for the UHRs, it would be the difference in rank between all the pairs. For the patient data, it would be biopsy 1 minus biopsy 2, taking the absolute value of that. From there, we would compare the UHR rank deltas with the rank deltas for the two patient samples. Now we can take a look at the impact of filtering on the hallmarks. So this first column here indicates the 27 gene sets that were prioritized by KDL. These next 4 columns indicates the filtering thresholds that we looked at. And then the one that we want to focus on is keeping genes with greater than 10 average expression, because we want to filter out the low expressing genes. And so this pathway right here, crash signaling downregulated, had over 93% of the genes lost when we're looking at keeping genes with greater than 10 average expression. And we can look at this even further, where there were 5 pathways with greater than 70% of the genes lost, and then only 2 pathways were less than 20% of the genes lost. So one of the pathways I'd like to look at is IL-6 JAT-STAT3 signaling. So one of the questions we had was, are the low expressing genes in the UHRs also low expressing in the patient samples? Because the whole idea is that we want our UHR reference distribution to be well representative of the patient distribution. And so we're seeing that this isn't the case, because the low expressing genes in the UHRs are both high expressing and low expressing in the patient data, meaning that they're not well representative of the patient distribution, and there's also variability in how these genes are being expressed in the patient distribution. And similarly, we can look at this in terms of ranks, because high expression genes would receive a small rank value, whereas low expressing genes would receive a large rank value. So there's variability in how the ranks are too. So as an overall, our main question with this AIM2 was to determine if the UHRs would give us an appropriate reference distribution. So as we saw, a number of the hallmarks would not be able to be evaluated given the percentage of genes missing if the UHRs are the reference. Normal tolerated missingness is 20%, but we had pathways where greater than 70% of the genes were lost. And secondly, of those who are low expressing in the UHRs, we don't see them as consistently low expressing in the patients, because again, the low expressing in the UHRs were both low expressing and high expressing in the patients. And additionally, we need to consider that some genes will be variable due to the disease status of the patient, not just looking at UHR versus primary sample tissue, but patient-specific considerations. In addition, the UHRs also appear to inflate patient outliers if low expression is not accounted for. And since we've been looking at this in the context of breast cancer, we can look at this in assessing other disease types as well. So again, taking the same pathway, IL-6 JAT-STAT3 signaling, we took these genes, we imported it into GTEx, and the GTEx project is samples collected from 54 on-disease tissue sites from about 1,000 individuals. And as we can see from this, when we're reading this plot, the yellow indicates that these are low expressing. The blue, the more intense the blue gets, that means that the genes are highly expressing. And so overall, we can see that the UHR distribution may not be expressing the relevant genes. And so if we can see this lime green section here, this is lung, we can see that if the UHRs were to be used as a reference distribution for lung, it would underestimate the majority of the genes. And so almost all the patients would be seen as overexpressed for this given pathway. And so going back to the key characteristics that we defined for an optimal reference or null distribution, we stated that the expression in samples should be appropriate for the disease type. And as we saw, this isn't the case, because the UHRs had a lot of low expressing genes. So that means we would have a loss of the hallmarks, and these aren't representative of other types. So for instance, like the lung example that we just looked at. The second characteristic that we stated was that there's biological variability sample to sample, but isn't due to technical artifacts. This also isn't met because we had batch effects. Lot 1 compared to lot 2, and lot 1 and lot 2 combined were variable. In addition, we said that there should be a large enough distribution to avoid sampling error. And so this isn't met as well because of batch effects, and then there's also issues of sampling genes dependent on the gene filtering criteria. And then the fourth characteristic that we stated was that pairwise differences among samples should reflect normal biological variability, but be less than the actual treatment effects if there are any present. And this would allow us to identify real changes on therapy. This also isn't met because we had batch effects, and then low expressing in the reference distribution led to artificial inflation of patient metrics. And so as an overall summary, in AIM-1, we looked at the evolution of testing, targeting the question of how has laboratory testing evolved. Then we looked at the increasing complexity in laboratory testing. How do we address the gaps in pipeline development? And in AIM-2, we transitioned to looking at an increased emphasis on rigor and appropriateness. So essentially, how do we validate models? How do we evaluate the appropriateness of said validation? Then we saw that there was an increased interest in a null reference distribution. And from there, the null reference distribution would give us a baseline point to determine if there was a real treatment effect. And then lastly, we looked at the increasing need for validation. How do we best evaluate the methods? And as an overall conclusion, to effectively transition from research to clinical usage, there is a great need for the validation and hardening of complex and heterogeneous algorithms. Thank you so much for your time, and I'll be happy to take any questions now.
Thank you. Very interesting talk. Are there any questions from the audience? You can either unmute yourself or type them into the chat. Actually, I can make a comment, and it has little to do with your work, which is outside my area. But as we see all these, and actually, maybe there's a potential angle as we see, all of the kind of AI models, and I guess some of which apply to clinical laboratory tests, but also apply to other kinds of data. Wondering whether we need some similar kind of regulation in AI. I mean, you can comment on it. I'm not really asking you a question, but the thought pops into my mind.
She kind of touched on that, but I think, Bill, you're exactly right. We're seeing that with the FDA, right? They're reconsidering the regulation now, right? So it's very timely.
Good. Well, okay. If there are no questions from the public, we can dismiss everyone, including myself, and leave Angie with her committee, and they'll take it from there. So thank you, everyone, for attending.
Thank you.